Hi class, in this lecture we are going to be looking at transcription. Now we are on the transcription. Transcription is involved with making proteins, and so this is done all the time. Replication only going to happen when the cells divide, whereas transcription is going to take place over and over again because you need proteins all the time. That is one of the significant differences between replication and transcription. This is the application of genetic code. So we are going to talk about how DNA is converted to RNA. When that DNA is converted to RNA, we call that transcription. So it is making a DNA to RNA copy. The next step is where you change that RNA language into protein language. There are two different languages. If we talk about the two biomoleculars, you have nucleic acid and proteins. Nucleic acids are read in one fashion, then proteins are read differently. We call that process translation because you are translating the RNA language to a protein language. This is taking genetic code and converting it into protein code. We'll look at all these different steps as we go along. We call this the central dogma of biology because this is so important for organisms to live. If you do not make proteins, you are not metabolizing. So you need to go and take the blueprint of DNA and convert that DNA to RNA, which I call worker. And then that worker will build the product, which is the protein. Each triplet of DNA or RNA represents a code for a specific amino acid. The protein's primary structure is the amino acid organization. That DNA tells you what the protein will look like by telling you what amino acids will go and in what order. And again, the proteins determine the phenotype. Living things are what their protein make them. DNA is the blueprint. It tells you what the cell will do, how it will function, and what it will make. So first, we need to understand what is RNA. RNA is a single-stranded molecule made of nucleotides. As you know, there are some differences between DNA and RNA. One difference is the sugar. Instead of deoxyribose, RNA has ribose. The other main difference is that instead of thymine, RNA has uracil. We have some different types of RNA. We are concerned with three of them. The first is messenger RNA, mRNA, which carries DNA message through complementary copy. The message is the chiplet called codons. It is going to transcribe the DNA code into RNA, and then that RNA is going to be made into protein. Transfer RNA, tRNA, is made from DNA. The secondary structure of tRNA creates loops. It has an amino acid attachment set at the top, and the bottom loop exposes a chiplet of nucleotides called anticodon, which designates specificity and complements mRNA. It carries a specific amino acid to ribosomes to build proteins. The last one is the ribosomal RNA, rRNA, which is the ribosome's component where protein synthesis occurs. Alright, so let us set up transcription, and this is the first stage of gene expression. Instead of DNA polymerase, now we are thinking about RNA polymerase. So we are going to make an RNA copy. It binds to the DNA upstream of the gene. We call that region promoter. RNA polymerase starts to add nucleotides built in 5' to 3' directions. RNA has uracil as atomized complement. It determines RNA polymerase recognizes signals and releases the transcript. Now it is ready to start the translation. mRNA tends to be anywhere from 100 to 1200 base pairs. Roughly around 500 base pairs is about the average size. That is depend on the size of the gene. Alright, so let us see some pictures and see if this makes any more sense. So the RNA polymerase is involved with the unwinding and building of the strand. We have two strands of DNA here. We have what is called the template strand and the non-template strand. The template strand is going to be used as template. How you build your RNA. It looks basically the opposite of the template strand because these are complementary. So the mRNA is the same as the non-template strand. The only difference is that instead of having T's put in, you now have U's. So here is the video. Watch the video and I will come back in and show you here in just a second.
The synthesis of messenger RNA is called transcription. Transcription begins when RNA polymerase recognizes and binds to the promoter region on the double-stranded DNA molecule. A particular subunit of the messenger RNA, called the sigma factor, participates in recognition of the promoter region. Soon after transcription is initiated, the sigma factor dissociates from the RNA polymerase. RNA polymerase moves along the template strand of the DNA, synthesizing the complementary single-stranded messenger RNA molecule. Synthesis is in the 5' to 3' direction, with new nucleotides being added to the 3' end of the growing messenger RNA molecule. As the RNA polymerase advances along the DNA, it melts a new stretch of DNA and allows the previous stretch to close. When RNA polymerase reaches a specific sequence of nucleotides on the DNA called the transcription terminator, a hairpin loop structure forms in the messenger RNA, causing the RNA polymerase and the messenger RNA to dissociate from the DNA.
I like this video because it shows you exactly what is happening here. Remember, mRNA has to be built in 5' to 3'. So what mRNA does is it reads the template and rebuilds an exact copy of the non-template, except replacing the T's with U's. Watch this video again if you need to.